So, welcome everyone, and thank you for joining us this afternoon. And thank you to the speakers. It's quite early for them. Today we have Volker, Gang, and Johan who will be talking to us about expansion and their experience. And hopefully we'll be able to learn a lot from all that they have done and the ways to improve the expansion protocols. So I'll leave you guys to it.
Yeah, okay. Good morning, good evening for you, I understand, in your time zone. So I'm Johan Hofkes from KU Leuven, chemistry department there. And today we will talk a little bit about what we have been doing in terms of expansion microscopy. So we divided the effort. I will give a quick introduction. Volker will show the chemicals that we have been developing, and Gang is here for any practical questions you might have. So our effort in expansion microscopy dates back to 2015, actually. I was at the International Conference of Photochemistry organized by Marcus Sauer in Würzburg, together with colleague Maarten Roefhuis. We were involved quite heavily in super resolution microscopy at that time. And that conference is the first time we have seen somebody talking about the expansion microscopy. And in all fairness, when I saw it the first time, I was very skeptical. Unlike my colleague Maarten, who immediately kind of embraced the idea of physically expanding your sample as a way to realize super resolution microscopy. Now, my reluctance came from the fact that I'd been working before with polymers. There's typically a lot of heterogeneity involved whenever you work with polymers. So I was very skeptical that this isotropic expansion that they claimed, whether that would be something real or not. And I still think that on a molecular level, this isotropic expansion might be debatable, and especially in view of the efforts that people are doing now to get nanometer or molecular size resolution in expansion microscopy. I still have some doubts that that is really reachable. Anyway. Despite the fact of my reluctance, we had seen an opportunity there to improve linkers, because like any super resolution microscopy method or any fluorescence microscopy method, more in general, hooking up your labels to the sample is a challenge that is always there. And we had been for years active in developing chemistries to get reliable labeling of your sample. And so that's where we come in then with the design of new linkers that are easily adaptable, that are universal for expansion microscopy. And actually, most of the ideas come from Volker. And I see a message that the slides are that's completely okay. Sorry?
I don't think your slides are changing.
Yeah, that's okay. I'm just talking without showing slides.
Someone was asking
so the real
yeah, okay.
I will stop. Stop chatting.
Sorry about
that. I'll give the floor to Volker. Anyway, I just wanted to describe how we came to this line of research. And in the meanwhile, yeah, it has been an active line of research where So far, 3 PhDs graduated, a few more on their way to graduation. And without further delay, I will give now the floor to Volker, and he will show what we actually have done in terms of chemistry and in terms of development of linkers. So Volker, go ahead.
Thanks, Johan. All right, hopefully slides move now. This moved. All right, so I will give a very, very brief introduction on how we got around to this topic. Johan already mentioned a bit of it. You know, when in high school, you always see these nice graphs of cells as you know, a soup of things where everything is floating around very nicely, freely, but that's really not true. These things are really dense, really compact, and there are many, many, many layers in there, as you all will know. You have DNA in there, you have RNA in there, you have proteins, all these things interact with each other, and getting that information out in, you know, a concise, understandable manner, that's really no easy feat. So we have been working in the Hofkes group for more than 10 years already on generating chemical tools, chemical concepts for analyzing omics data that can go from, we have a big branch of metagenomic analysis technology. And then this is actually a project here where we're talking about today about new ways of uncovering cellular information in a way that the context of that information is not lost. And as Johan mentioned, fluorescence microscopy, high resolution fluorescence microscopy is always our go-to tool for analysis. So the way I see that expansion microscopy came about, and this is totally, you know, unfounded, is that probably when around 2012, 2013, the existing clearing methods emerged into the Clarity protocols. Someone at MIT must have been doing this and, you know, got annoyed by his gels swelling. And that's probably where this nifty little invention was made of using this swelling to get to expansion microscopy. And indeed, as Johan mentioned, we saw this first in Würzburg in Germany back in 2015 when this was very fresh. And we said, Johan didn't say at the time, but we said, well, this could be something where our linker work is usable. And we then started thinking about combining functions into single linkers where you have the targeting moiety. Let's see if I can get some kind of pointer on here. Yeah, where you have a targeting moiety, where you have a reporter moiety, and then this all can be grafted to the polymer. And this would have the benefit of simultaneously locking the position of the biomolecule of the target and then locking it for analysis. And we had these ideas quite early on, but you know, it takes a little bit of time to go there in academia. But this is actually the overarching concept. What you will see in the presentation that follows is that we will talking a lot about this part here, about linkers. And we always will address a biomolecule. We will combine it with some kind of signal. This can be a fluorescent dye, but it can also be a hapten, or it can be a chemical reactive group. And then we have something that allows grafting to the hydrogel matrix, to the polymeric matrix. And then, of course, you all know expansion microscopy and how that would work. Now, what is important here is that it's covalent linking. All these things are covalent, so they are really stable. You can have the gel, and the gel is in principle forever stable. And our view is that ultimately we want to convert the entire biological information, relevant biological information, into something which is covalently grafted, stable, and you no longer worry about the biology because you have something which is a stable model, and you can interrogate this through cycles in a multiplexed manner. So back in 2018, we ran first tests of this, and our design was converted into first chemicals. I've tried to keep the chemicals a bit low. By the end, there is a bit more chemistry, but for now, this should be quite high level. And what you see here is a dye. This is what I mean 6G dye, which is a very nice fluorescent dye. We have an active ester for amine targeting, and we have a monomer. And this is actually the most simple variation of our multifunctional linker that you can devise. This one will graft to the polymer matrix during the acrylate polymerization. This you can attach to the antibody, and you will have your signal. So what does that look like? You take a secondary antibody, you incubate this with this reagent, and you then do your immunostaining. And these were the first examples of our reagents actually working. On the top, you see pre-expansion, and on the bottom, you see expanded samples. These were the first examples, and this was done by Donato, who joined us back then. He then moved on to a PhD project in Johan's group on perovskites. He looks a lot scruffier now. He should have stayed with us. And we continued working on that with Gang joining the efforts. And, you know, we made a bit more complex things. We extended the linker, make a bit more water solubility in there. But in all, the design remains the same. We always have the signal, the reactive group, and then the monomer. And actually, and I can say this quite confidently, these things are quite easy to use, especially for antibody labeling. You take the reagent, you incubate it with your antibody, and you're basically good to go. It takes a few minutes, half an hour of incubation time. You don't even need to purify in most cases, and you just can move directly into staining. And, you know, we moved to primary antibodies as well. What you see here is a tubulin staining, which we use as a model system a lot, or laminin staining with primary antibodies, pre-expansion always on the left, and then post-expansion images on the right. We also made variations on this theme where we do not take a dye, where we convert the dye into a functional group. We exchange it for a functional group like this azide moiety here. But in all, the design always remains the same. You have the monomer group, you have a reactive group capable of linking to the antibody, but in this case, you have the azide, which allows you to take an antibody, you react it with the linker, you get the reactive antibody, which you can then polymerize into your expansion gel, but this azide moiety here will remain there, dark at that time, and then once your gel is formed or expanded, you can always add a dye, and you can generate your signal. And what's the main advantage here is that you can get access to dyes that would otherwise degrade in expansion microscopy. I'll come back to that dye effect a bit later in the presentation. We have made various reactive group systems here, azide, tetrazine, or biotin systems, and they're all doing the same. The biotin system, for example, allows you to pre-link your antibody with a biotin here. You then gelate everything, and then when your gel is stable and expanded, you add the antibiotin antibody, and you can start looking for your signal, which you introduced beforehand. We also built on this with oligonucleotide signaling, where the dye or the reactive group is exchanged for an oligonucleotide. Again, you take an oligonucleotide, you react it with one of our linker systems, and you get a multifunctional oligonucleotide, which is ready to react with an antibody, and you can do hybridization of your antibody and thus see your fluorescent signal. As always, the linker scaffold remains the same, the design remains the same, so very little changes there. And this can all be done from a very limited library of reagents in the end. And what you see here is then, again, anti-tubulin staining of an antibody, which was appended with an oligonucleotide. The length of these oligonucleotides can range from 15 to 30 base pairs on hybridization, depending a bit on what type of binding you want. And this all quite readily, quite out of the box. And of course, you have many variations on the scheme. I will not go into details. If you have technical questions, you can always come back to us or ask at the end of the presentation. There are small variations on how to do this, and they have an impact on your protocol. So if you, for example, take an antibody and you react it with our multifunctional linker, in this case, there's an alkyne reactive group here, you can choose to link the antibody with one of our linker systems rather than doing the oligo as you saw in the previous slides. And then you would only have to buy the oligonucleotide with an azide group pending. And this is commercially available. And in this case, you would not have to make fluorescent oligonucleotides. You can just simply buy them from your oligonucleotide supplier. The linkers used in this case are always built on the same thing again. But here you see that you have a DBCO group, which is a cyclooctane group, that will allow very mild linking of the fluorescent stain upon hybridization of your system. Again, pre-expansion, post-expansion shows a very nice contiguity of the tubulin signal in this case. And then a final variation of this scheme is grafting the docking probe. You have an oligo-appended antibody. You have non-functionalized oligonucleotides. But in this case, you have a fluorescent multifunctional linker. You have, again, a rhodamine 6G dye here on top, the left. And then you have, in this case, 3 acrylates. I will come back to why that is later. And in this case, we have a reactive group for nucleic acids. So this is a platinum complex that will graft itself to the backbone, to the guanine basis of this oligonucleotide, and then do backbone staining. And what's the advantage of such a system is that this is plug and play for your current procedure using oligonucleotides. You have an antibody with an oligonucleotide using your preferred method. You just buy the non-modified oligonucleotide matching to your barcode, and you just react everything with one single linker. Again, pre and post images in expansion of the tubulin system. And again, by simply switching out the dye for a functional group, you can go back to color switching. And then this is ATO 488 in green here, and this is Psi5 on the bottom. And again, Psi5 is a dye that is normally not accessible in standard expansion microscopy due to its full degradation in the radical polymerization. Can you build on these things for super resolution methods such as DNA paint? Yes, you can, but it's not optimized yet. So what you see here is actually you take an antibody which has a short oligonucleotide appended to it, and then you have another short oligonucleotide as a docking sequence coming in, but the binding is transient. And because this binding is transient, the dye will only be at the location of the oligonucleotide for a limited time, but by accumulating this signal for a longer period, you will get super resolution imaging. And this is what you see here. But as you can clearly see, it's unoptimized due to the background. We add a certain amount of imager strands in this case, and that will cause some background signal. What you see here about signal to noise ratio of 6, which we still aim to get higher. We're not really actively pursuing this, but it remains an interesting application that can be developed. So from moving on from the standard immunofluorescence approaches and the antibody staining, we then sought to extend the biological information that we could address. And this started first with phalloidin staining for actin staining. So the actin monomer is actually a tetramer, which has 4 clefts, where phalloidin is a very strong binding ligand. And this is used by most commercial phalloidin stains for actin. There are a few other ligands, but they're not used as widely. So what we did is we switched out our biological targeting moiety for phalloidin, the design always remains the same. We have again a dye. In this case, this is a rhodanine B dye, which is a bit more redshifted than 6G. And again, an acrylate monomer for matrix grafting. And actually, from the first time we tried it, this worked nicely. These are the first images back in 2019. PA expansion and post-expansion, where the actin filaments were visible. This actually turned out to be a very interesting probe, where many people have seen use. So we started to optimize this a little bit. This also allowed us to gain some insights in what actually the structural requirements are for a linker in expansion microscopy. Here you will, on top, you will see the most basic systems. You will see a dye, rhodanine B, and phalloidin, but no acrylate. A variation on this theme is our multivalent linker, as shown in the previous slide. You have a rhodanine B dye, you have a phalloidin targeting moiety, and you have the acrylate. So this would be our core multivalent system. And what you see on the bottom here is, if you look at the signal from those two probes in actin staining, you will see that reagent 1 with ACX added for non-specific grafting of the actin proteins will give you some signal, but very little. When you introduce the acrylate, so you make a multivalent linker, this immediately jumps. What this shows is that actually the multivalent linkers do have an edge. And this is still the best method in literature out there for actin staining. We also elaborated on the ligand scaffold itself by making, for example, this triazine system. This allows a bit for, this is a chemical concept, convergent synthesis of all functional groups where you have one central core unit pasting everything together. They also work more or less the same as the linear system here, but we don't use that a lot anymore for synthesis reasons. These are easier to scale up at the moment and have a bit better water solubility. And then on the right here, you see how we can start making things where you have a functional group here, which is a maleimide, and your dye is only introduced at a later stage. This would be an easy way of making multiple linkers from one central molecule with a thiol addition to the maleimide. And you see that they also work slightly less, and that's probably because this bond here will not be super stable in the acrylate polymerization process. But in all, the linkers seem relatively robust in making actin signals, and we do need the acrylate system. So further optimization, moving from the system with one acrylate to multiple acrylates. And what does that buy us? Here you see one acrylate moving to two acrylates. There is an effect, but it's not huge. But when going to 3 acrylates, you see a significant jump. This probably relates to incomplete grafting of the polymer process. In the drawing, it's always nice to show that all acrylates in your mixture will be grafted to the polymer gel, but obviously that process is not perfect. And that's what's seen here. By simply increasing the number of acrylate groups on your linker, you will ensure that your ligand is grafted to the matrix and you will get an improved signal. Now on the right here, this is an interesting thing. You see these reagents, but then with the addition of ACX. And what's the message here is that ACX addition always helps. This is something that we have seen in all our experiments. We've also seen it in literature and from competitors. ACX, for some reason, always increases your signal. Probably here in the case of the actin system is that after grafting, you will get proteinase K step, for example, to digest your actin structure. But some protein fragments will remain, and these are then grafted by the ACX and further help in retention of the signal. The exact mechanism is not yet understood. Building on these reagents, we see that labeling is actually very reliable. And so far, actin staining can be conducted under all the expansion protocols tested. And here you see, for example, 4-fold standard expansion systems for the actin stains. And this is then 10-fold expansion using the T-Rex protocol. And you see, of course, these are then the attained resolutions after expansion, going to more than double increase for the 10x resolution expansion. We also have for the actin systems where there is a reactive group, as seen here. This is again the A side. Back to our design, we have phalloidin. We have groups capable of linking to the acrylate, and we have the A side here capable of linking to a cyclooctane dye. So what you do is you start by incubating your sample with this reagent. All actin gets tagged by the phalloidin. These reactive groups get grafted in your polymerization. And once your gel is stable, you just add the dye, again, Atto488 here on top and Cy5 on bottom, pre and post expansion. And again, Cy5, sulfo-Cy5 in this case, something that normally would not be surviving the expansion process itself. We can combine this with immunostaining. Here you see two examples of actin stain combined with antibody staining. These are the previous microtubuli secondary antibodies, and this is TOM20 against mitochondria. And you see that single protocols allow for multiplexed imaging with antibodies and the small molecule ligands for phalloidin. Again, pre and post expansion imaging. You have this sample now, which is stable. You have the signal incorporated into the matrix, so you can go for other ways of doing your imaging. And this is, for example, a SIM imaging experiment where you can clearly see the resolution gain, the additional resolution gain after expansion when you take your wide field image versus a SIM image of the expanded sample. And there are a few variations of this theme out there in literature with other microscopy modalities, but the ligands are amenable to all of them. This is, for example, some tissue experiments done by Kang with collaboration in Würzburg University, where you see tissue samples and you see neuronal samples, pre and post expansion. Here you also see antibody staining at the same time. And what you can see nicely here is that you can see the structure of the actin filaments resolved at these very high resolutions. And this is then the combination of the actin ligands with other microscopy modalities, but you should also take into account that by just pushing the expansion factor, you will also get similar information to this. These are our ligands, courtesy of Robert Miskier in Yale, at 16 times expansion, where you also see the very nice periodicity in the actin filaments. Some more examples from external users to show what is possible with these reagents. Here you have sprouting actin filaments using STED microscopy by Eugene Katruka in Utrecht in the Netherlands. These are networks of aggregated platelets and their actin framework by a collaborator in the UK. What you see here is images from a collaborator in Finland, showing nicely actin skeletons in inner ear tissue, and this combined with immunostaining here on the left. So in all, we have been sending these things out into the real world, and we're getting very, very nice feedback on their performance. So can we take these learnings and do some other ligands in small molecule ligands for the cell? The answer is yes, but it's not so clear-cut as with the actin. Because you would say, let's simply exchange the actin for a taxane derivative. So as in this case, you have paclitaxel, or here you have docetaxel. And again, you have the monomer, you have the signaling moiety here, in line with our overarching design. You add it to the cell and you expect to see microtubuli. And yes, that's true, but it's not clear-cut. What appears to be the case is that the positional arrangement of the ligand on the taxane derivative, so whether your linker is at this side of the ligand or somewhere over here, has a huge impact on the performance. So this remains to be optimized, but also the cell cycle, the cell type, and a big buffer dependence make this a case that is not clear-cut yet. We're still pursuing this, but it's not finished yet. Moving to other biological systems, membranes. We were the first one back in 2019 to show that you could address membranes with ligands. Here you see pre-expansion. Everything is a bit stained and blurry, but after expansion, this clears up nicely. You can clearly see double membranes, sprouting vesicles here, and then onto the design. How is that done? Again, multifunctional ligands. You have a dye here on the left, you have a monomer here on top, and you have a lipid in this case as the biological targeting moiety. These are some literature examples of people who have described similar systems, just for your reference. This is our system, where you have a Pacific blue dye in this case. And this is the image I just showed before. Again, a lipid here, a linear linker system, multivalent here, and then biotin for staining afterwards by Garigianis in 2019. And this can be used to stain lipids by simply including ACX. And then from Germany, there is this simple sphingolipid linker, which also using glyceraldehyde and staining on the A side afterwards can provide you with lipid staining. Here, this is an example of a multivalent linker, which is for a gel system that is not acrylate-based. Does it work? Yes, it works, but some care needs to be taken in what system you use. For example, if we switch out the dyes, Pacific blue, shown here at 405 nanometers, is a very general lipid stain. It will stain all your membranes, and it will be quite cell permeable and can be used for general staining of systems. But if you go for some of the other dyes, as mentioned before, ATO488, rhodamine 6G, or rhodamine B, you will see that the dye influences the behavior of the multivalent linker. In these cases, which are 6G and B, rhodamine 6G and B, you will see that they get some tendency to aggregate in the mitochondria, and that's due to their charged dye. The quick solution to this, which is not shown on the slide, is exchanging again for an azide. You get a neutral system and you get broad staining. What is also not shown, but what we have done, is exchanging this lipid for other lipid types like cholesterol or sphingomyelin, and then you get different kind of behaviors. These are not really studied to the full extent yet. Then we moved on to nucleic acid grafting. There were a few examples in literature quite early on. For example, this Tsanov here, or the original paper from Chen, 2016. What they do is they graft the RNA, in this case here. So they have an RNA binder for grafting the RNA to the matrix. Tsanov here used an approach where they have a secondary binder that can be grafted to the matrix. But what we said is, let's bind the docking probes. Why? Because they will find your biological information and then be bound to the matrix covalently, and they will remain there forever. And then you're not depending on the length of the RNA or the structure of the RNA. So we tried to do this, and we came up with our design consideration. Again, a fluorescent reporter. This is the same molecule as you have seen before. Fluorescent dye, 3 acrylates in this case, and then a reactive group for nucleic acids. We've modified a lot this reactive group for nucleic acids, trying to find the one that's most performant for targeting the nucleobases. We've looked at probes that can be activated by light or by probes that can be activated by heating. As you, for example, see here, this platinum complex as seen before, or a nitrogen mustard. What you see here is a psoralen ligand, which also works very nicely, and some other light activated linkers. So the final probes look a bit like this. You have, again, a fluorescent dye, an acrylate, and one of the reactive groups. And then what we did is we took an antibody with an oligonucleotide on there, and then we labeled the complementary oligonucleotide with these probes. And then by looking at the hybridization, you get simultaneous info on the binding, but also on the efficiency of the labeling. And then what you see from this is that you can get to very high labeling densities here, while still maintaining very efficient docking to the target probe. And you can get very clear signal. So in all, these are reagents that can be used for the very simple creation of labeled DNA, labeled RNA. They do work best on single-stranded systems. That's just their chemical nature. And we then move these things into in situ hybridization systems, with the most easy approach being simple RNA in situ hybridization, RNA fish. And then we extend it into whole fish and HCR for amplification means. So this is a slide comparing our approach to what other people have been doing out there in literature. This is the original system from the Boyden group, which is label X. This is created by making ACX here on the left and letting it react with label it, which is a commercial reagent from Myers Biosciences. And they reacted in situ to generate this compound. It's not isolated at the time. A recent paper by Wang describes how the Melfa X variant, by using not this simple nitrogen mustard here, but using the double nitrogen mustard, has improved binding efficiency. We tested this, the variant of this where the carboxylic acid here is not present, and that works at least equally well, so it's not needed for the binding. It's probably mostly to do with the fact that there's here double nitrogen mustard, which increases the activity. And then this is our prime system, platinum-based and 3 acrylates. And so when using these things in plain old RNA fish, you have a target RNA, you buy some complementary docking sequences. These are DNA oligos, and you let them react with the reagents from the previous slide. You just heat it briefly, or if you want to use the light activated versions, you submit it to UV activation. And this gives you the probe library with a fluorescent dye on there and with an anchoring moiety here. And they will provide you with fish. What you can see here is a housekeeping gene in HeLa with fish. What is not shown on here is that this actually needs quite a bit of laser power to give you a good signal. The signal is diluted. There is only one dye on there. So amplification is something that would be nice to have in these cases. Especially, the housekeeping gene is quite abundant, but if you want to go for low copy number things or very sparse RNA, then you would like to have some kind of amplification. So we move this to rolling circle amplification. Rolling circle amplification, I won't go into details, but this is basically a padlock probe that you add, and then you start doing an amplification of a continuous bundle of DNA, a Roloni, and this can then be stained fluorescently and to give you a signal. And here is an example of GUS-B in HeLa with its after expansion with its negative control. And you can see nicely clear spots of an RNA that is less abundant. What should be noted here is that all these things require secondary hybridization. So you have a system where everything is grafted to the gel, and then you want to have an oligo docking to another oligo. But these oligos don't like to dock at low salt concentrations. And this is actually what you need for expansion. So in all these cases, you should take into account that there is a hybridization buffer needed to allow for efficient signal generation. And this means that you will probably need to have a secondary polymerization of your sample to get something that is stable enough to go through these hybridization steps. This is all somewhat optimized in some of the papers that we did, but also in recent work of the Boyden group, for example. Another signal amplification approach is the HCR, hybridization chain reaction, where you can nicely see two targets at the same time. What should be noted here is that you have the third generation HCR. We have our linkers linking the error correcting probes to the target, to the matrix at the same time. And then you have probes that will concatenate and generate the signal. And in this case, you see two targets at the same time, one very abundant and one less abundant signal, and you can clearly pick them up. So this is a first example of multiplexing for RNA with our ligands. And you can do similar things with antibodies. If you have a low abundant antibody, in this case we use tubulin, which is of course not low in abundance, but shows the purpose of these reagents. This is the antibody with a single docking sequence strand, one dye on each strand, probably a few on each antibody, 2 or 3, and you see a dim signal. But if you take the probe and you have concatenating complementary probes, you can start generating this longer concatenated product, and you get immediate signal increase. This is a Saber-like approach, and that means that what I want to show here is that the multivalent linkers of us directly allow the use of these Saber approaches for antibodies as well. All right, and then finally, a little bit more chemistry, and I want to talk a few more minutes still about the linker itself. So I've shown how the linker can be used to target various biological molecules, moieties, lipids, actin, antibodies. But the linker itself is not without use. On the left here, you see the one that is used most, which is ACX. What we've done is we've made variants of this with improved coupling efficiency. This is a tetrafluorophenol ester, which is more reactive and more water stable, so it will not hydrolyze as quickly when you're tagging your antibody or your proteins. We've also made variants where you have 3 acrylates. I've shown you that this will get you better grafting to the gel of your target, especially when you have something where there's only one linker on there. We've also made linkers for direct nucleic acid targeting. This is, for example, one of the linkers used in the ISH approaches. Here you can see a charged backbone. This charged backbone linker will allow for charge effects. This positive linker will like the negatively charged nucleic acids, will ensure fast binding and very efficient grafting. And then these are linkers where we do direct hapten inclusion. You can see a linker here with a biotin or a DIG hapten And these can be used for secondary labeling approaches. You take one antibody, for example, and incubate it with a linker that has a biotin. You graft it to your matrix, and from then on, you will have at the position of your antibody a biotin that will be forever there for interrogation with an antibiotin antibody. So these are simple linkers, and we sought to extend them into something which can help in the dye stabilization, because actually dye stabilization is very relevant to expansion microscopy, because dyes don't really like radicals. And this is a figure showing how dyes degrade over the process. You can see that a few survive the process, but other dyes like Cy5 are totally killed. There's very sound chemical reasoning behind this. It has to do with double bonds on the system themselves that do like the polymerization process themselves, and they will polymerize and lose their chromophore. So we thought you have similar concepts in dyes, where you have a dye that can be self-healed by having something that can quench the radical or capture the radical and recover the dye. I will quickly skip through this. You can look at it afterwards if you want to see some more thing, or refer to the manuscript from Gang here. But what does that look like? We take dyes, various dyes here, and we attach a stabilizing group. For example, in this case, nitrophenolalanine here. In case that the dye gets attacked by a radical, this group has the capability of taking on that radical and restoring the dye. That was the postulation here. So what do we see here? You can see that in all cases, ATO488 or the stabilizer appended, you will get a signal. Sometimes the signal is higher for the NPA. Sometimes, in most cases, the signal here will be higher for the NPA. It's a bit unclear here. Cy5 still gets degraded a lot, but it is higher. Now, this is the signal retention, which is measured by dunking these things into a gel, washing that gel, and seeing how much fluorescence remains. So there's always more fluorescence when you include this NPA. However, it's not a straight cut story, because if you start looking at absolute quantum yields of these systems, then you see that there's a trend. The unmodified system, which is a dye and acrylate linker, multivalent linker into that gel, will have a high quantum yield. When you go to the polymer, the quantum yield of the dye actually drops. When you then take the NPA, so the stabilized dye, into the milky water here, the quantum yield is always lower. Here it's near zero, but it's always lower than the regular one without the stabilizer. But when you then go into the polymer, the quantum yield actually increases again. So what this shows is that there are some crowding effects and some quenching effects that are not yet fully understood. But do they work? Yes, they work, and they have some interesting effects. What you can see here is one of our full scaffolds. This is then a tetravalent linker. You have a dye here, you have the stabilizing moiety here. Again, something to link to antibodies and a polymerizable group. You can make an antibody with this multivalent linker, and you will see again very nice tubulin staining before and after expansion. And when you then look at the signal, you will see that the signal pre-gelation is actually lower, but after gelation is higher. And this is then probably related, in our current view, to the increased quantum yields of the dye after expansion, where you decrowd the dye a little bit. Here you see that there's also an effect on photostability here. Overall, the NPA linkers allow longer imaging. The dye remains more stable. And then we made a few things, a few other variants here. What you can recognize here is, for example, another stabilizing group Trolox to counter versus the NPA here. And we made linkers to them. We added them to antibodies, and you can see that they all have some effects, but it's not all beneficial. What you do see here, again, better survival, but the retention seems to be slightly lower. Why is that? That really does not make a lot of sense, because in the end, the chemical groups are all the same. So this is something that we don't really fully understand. There's probably some kind of photochemical, photophysical effects playing here that we don't really fully understand. But in all, I want to conclude here to show that the linkers by itself are not innocent, and that they can be used to exert effects on the signal, but that this still requires a little bit more study. So, concluding, over the past 5 years, we've shown that we can address a lot of biological information by simply using multifunctional linkers. It's currently the best option for actin and lipids staining in expansion microscopy. We show that they have improved signal retention versus the other methods out there, regular antibody staining. And overall, the protocols are very simple. Just dunk the reagents with your antibody or with your nucleic acid, and you should be ready to start your expansion microscopy experiment. And they also work over all the different expansion modalities, and there are by now quite a lot of those. What are we still doing? We want to get this into a single approach for oligo tagging, where barcoding the signals is probably the way forward. We take out the dyes, and we can have many, many targets in a single blueprinted biological sample. What else? We're also extending into non-radical based gel formulas, but that's not finished yet. And something that I briefly highlighted, but that's very annoying. We don't understand fully yet why ACX always helps, and it would be great if we could have some better understanding. Here in the bottom, you can find the references of this work, mostly work done by Gang. And I would like to conclude by giving a big thanks to all those who helped over the years. There were many of them. And should you have questions, you can always reach out to us. And this is probably a brief description of to whom you should reach out with what type of question. All right, and that leaves me with a few more minutes for questions.
Thank you for that. That was great. We'll keep a few minutes. If someone wants to write up a question in the chat, I can read them for them, or if someone wants to unmute themselves, feel free to do that. I think we already have a question from Hamid. He's asking if it's possible to develop a live cell expansion microscopy technique, possibly in conjunction with superfast light sheet microscopy that enables real-time dynamic imaging of expanded cellular processes with living organisms, and what are the key scientific and technical hurdles of achieving this goal?
Yeah, that's a question that we've seen a few times. Yeah, you need to polymerize your sample to get it to expand. So far, we have no answer to this. Yeah, and that's basically the brief answer. It would be amazing if we could do it, but we at the moment have no answer. Yeah, your cell would have to survive this in a way. Yeah? You need to get monomers into your cells, or you need to find a way to polymerize your entire cell while maintaining functionality. It's a really nice idea, but I have no immediate answer. Sorry for that.
One thing I was wondering is about the size of these linkers. How do they affect, like, how would they affect the expansion or like the distance that you're measuring?
Very, very little. But this depends a bit on the target you're doing. If you're using antibody approaches, then of course you have the size of the antibody that's always impacting your positional accuracy. And especially if you have then a sandwich where you have a primary and a secondary, this will incur quite some error. But when you're looking at, for example, RNA, these things are really linked at the position of the RNA. So there the positional error is quite low. The linker itself, when compared to an antibody, is tiny.
We have a question from Stefan. It's asking if there are linkers for nanobodies instead of the large antibodies, and how can he get hold of the reagents?
So all the reagents that were described in the talk as working for antibodies will directly transfer to any protein system. We have several ways of tagging them. You could tag them through tiols, or you can tag them through amines. We also have post-staining methods for proteins. Not all of them were shown in the talk. And the reagents are available from us for research use and testing, but also commercially available. I must say that Australia is quite peculiar to ship samples to and ship reagents to its customs, but we have done so that's no big issue. So if you're interested in something in particular, do reach out to us.
Having said this, Volker, I don't think we ever worked ourselves with nanobodies. Huh?
No, we ourselves have not worked with nanobodies, but I do know of people who have.
With our reagents.
Yes, with our reagents.
Yeah. In principle, there should be no issue to translate all this from antibodies to nanobodies. Yeah. There might be some peculiarities that need to be fine-tuned or so, but it should work straight away.
Yeah, one small remark that I do want to make here, I did not show the information here, but there is, of course, a question always with these nanobodies and antibodies. Are you killing their specificity by putting in a lot of linkers on them? And we have some data that shows that if you really decorate your antibody with a lot of linkers, then you're bound to have some of these linkers interfering with the binding site of the antibody. So, that's something that you do have to take into account, that you might have to optimize this a little bit for your particular application.
Any other questions? One thing that I have a question might be a bit more related to the microscopy side. Like when you are doing this imaging the same cell that is pre and post expansion, do you have a way of going back? How do you go back to the same cell to?
Gang, maybe you can answer this best. Gang?
Hello, can you hear me?
Yes.
Yeah. Okay. Yeah, it's always tricky to find the same cells back, especially if you expand your sample 10 times. But indeed, there are some ways. For example, when you just perform the pre-expansion imaging, you just mark a circle where you image your sample, and then you perform the gelation. After gelation, you of course just cut the area that you pre-imaged, and then you expand it, and after expansion, you just screw the area and find the same cells back. In my case, nucleus is always a good reference to find the same cells back, because nucleus can be strongly stained with the DAPI, and DAPI is very strong against the polymerization step. So you can always find the nice signal of DAPI after expansion. And of course, for nucleus, if the DAPI signal is lower, you can restain with DAPI after polymerization. And actually now recently there is another paper, the technique is called gel map, and they just on your sample, they create a fluorescence grid, and they can use the grid to find for example, to find whether it's isotropic or something else.
Thank you. If there's no further questions, just thanking you guys for your presentation today, or waking up a bit early. We already have scheduled a next meeting, which is going to be on the 29th of January. So, yeah. Thank you very much, and see ya.
Okay, thank you.
Bye.
Bye-bye.